4.4 Protein denaturation and folding. Proteins lead a surprisingly precarious existence. As we have seen, a native protein conformation is only marginally stable. In addition, most proteins must maintain conformational flexibility to function. The continual maintenance of the active set of cellular proteins required under a given set of conditions is called proteostasis. Cellular proteostasis requires the coordinated function of pathways for protein synthesis and folding, the refolding of proteins that are partially unfolded, and the sequestration and degradation of proteins that have been irreversibly unfolded or are no longer needed. In all cells, these networks involve hundreds of enzymes and specialized proteins. As seen in Figure 4-25, the life of a protein encompasses much more than its synthesis and later degradation. The marginal stability of most proteins can produce a tenuous balance between folded and unfolded states. As proteins are synthesized on ribosomes, Chapter 27, they must fold into their native conformations. Sometimes this occurs spontaneously, but more often it requires the assistance of specialized enzymes and complexes called chaperones. Many of these same folding helpers function to refold proteins that become transiently unfolded. Proteins that are not properly folded often have exposed hydrophobic surfaces that render them sticky, leading to the formation of inactive aggregates. These aggregates may lack their normal function, but are not inert. Their accumulation in cells lies at the heart of diseases ranging from diabetes to Parkinson's disease and Alzheimer's disease. Not surprisingly, all cells have elaborate pathways for recycling and/or degrading proteins that are irreversibly misfolded. Figure 4-20. The transitions between the folded and unfolded states, and the network of pathways that control these transitions, now become our focus. Loss of protein structure results in loss of function. Protein structures have evolved to function in particular cellular environments. Conditions different from those in the cell can result in protein structural changes, large and small. A loss of three-dimensional structure sufficient to cause loss of function is called denaturation. The denatured state does not necessarily equate with complete unfolding of the protein and randomization of conformation. Under most conditions, denatured proteins exist in a set of partially folded states. Figure 4-20. Denaturation. Most proteins can be denatured by heat, which has complex effects on many weak interactions in a protein, primarily on the hydrogen bonds. If the temperature is increased slowly, a protein's conformation generally remains intact until an abrupt loss of structure and function occurs over a narrow temperature range, Fig. 4-26. The abruptness of the change suggests that unfolding is a cooperative process. Loss of structure in one part of the protein destabilizes other parts. The effects of heat on proteins can be mitigated by structure. The very heat-stable proteins of thermophilic bacteria and Archaea have evolved to function at the temperature of hot springs, 100 degrees C. The folded structures of these proteins are often similar to those of proteins in other organisms, but take some of the principles outlined here to extremes. They often feature high densities of charged residues on their surfaces, even tighter hydrophobic packing in their interiors, and folds rendered less flexible by networks of ion pairs, which make these proteins less susceptible to unfolding at high temperatures. Proteins can also be denatured by extremes of pH, by certain miscible organic solvents such as alcohol or acetone, by certain solutes such as urea and guanidine hydrochloride, or by detergents. Each of these denaturing agents represents a relatively mild treatment in the sense that no covalent bonds in the polypeptide chain are broken. Organic solvents, urea, and detergents act primarily by disrupting the hydrophobic aggregation of nonpolar amino acid side chains that produces the stable core of globular proteins. Urea also disrupts hydrogen bonds, and extremes of pH alter the net charge on a protein, causing electrostatic repulsion and the disruption of some hydrogen bonding. The denatured structures resulting from these various treatments are not necessarily the same. Denaturation often leads to protein precipitation, a consequence of protein aggregate formation as exposed hydrophobic surfaces associate the Aggregates are often highly disordered. The protein precipitate that is seen after boiling an egg white is one example. More ordered aggregates are also observed in some proteins, as we shall see. Amino acid sequence determines tertiary structure. The tertiary structure of a globular protein is determined by its amino acid sequence. The most important proof of this came from experiments showing that denaturation of some proteins is reversible. Certain globular proteins denatured by heat, extremes of pH, or denaturing reagents will regain their native structure and their biological activity if returned to conditions in which the native conformation is stable. This process is called renaturation. A classic example is the denaturation and renaturation of ribonuclease A, demonstrated by Christian Anfinsen in the 1950s. Purified ribonuclease A denatures completely in a concentrated urea solution in the presence of a reducing agent. The reducing agent cleaves the 4 disulfide bonds to yield 8 cis residues, and the urea disrupts the stabilizing hydrophobic effect, thus freeing the entire polypeptide from its folded conformation. Denaturation of ribonuclease is accompanied by a complete loss of catalytic activity. When the urea and the reducing agent are removed, the randomly coiled, denatured ribonuclease spontaneously refolds into its correct tertiary structure, with full restoration of its catalytic activity, Fig. 4-27. The refolding of ribonuclease is so accurate that the 4 intracane disulfide bonds are reformed in the same positions in the renatured molecule as in the native ribonuclease. Later, similar results were obtained using chemically synthesized, catalytically active ribonuclease A. This eliminated the possibility that some minor contaminant in Anfinsen's purified ribonuclease preparation might have contributed to renaturation of the enzyme, thus dispelling any remaining doubt that this enzyme folds spontaneously. Figure 4-27 denatures the ribonuclease, thus cleaves the disulfide, involves The Anfinsen experiment provided the first evidence that the amino acid sequence of a polypeptide chain contains all the information required to fold the chain into its native, three-dimensional structure. Subsequent work has shown that only a minority of proteins, many of them small and inherently stable, will fold spontaneously into their native form. Even though all proteins have the potential to fold into their native structure, many require some assistance. Polypeptides fold rapidly by a stepwise process. In living cells, proteins are assembled from amino acids at a very high rate. For example, E. coli cells can make a complete, biologically active protein molecule containing 100 amino acid residues in about 5 seconds at 37 degrees Celsius. However, the synthesis of peptide bonds on the ribosome is not enough, the protein must fold. How does the polypeptide chain arrive at its native conformation? Let's assume conservatively that each of the amino acid residues could take up 10 different conformations on average, giving 100 different conformations for the polypeptide. Let's also assume that the protein folds spontaneously by a random process in which it tries out all possible conformations around every single bond in its backbone until it finds its native, biologically active form. If each conformation were sampled in the shortest possible time, 10-13 second, or the time required for a single molecular vibration, it would take about 1077 years to sample all possible conformations. Clearly, protein folding is not a completely random, trial-and-error process. There must be shortcuts. This problem was first pointed out by Cyrus Leventhal in 1968, and is sometimes called Leventhal's paradox. The folding pathway of a large polypeptide chain is unquestionably complicated. However, rapid progress has been made in this field, sufficient to produce robust algorithms that can often predict the structure of smaller proteins on the basis of their amino acid sequences. The major folding pathways are hierarchical. Local secondary structures form first. Certain amino acid sequences fold readily into alpha helices or beta sheets, guided by constraints such as those reviewed in our discussion of secondary structure. Ionic interactions, involving charged groups that are often near one another in the linear sequence of the polypeptide chain, can play an important role in guiding these early folding steps. Assembly of local structures is followed by longer-range interactions between, say, two elements of secondary structure that come together to form stable folded structures. The hydrophobic effect plays a significant role throughout the process, as the aggregation of nonpolar amino acid side chains provides an entropic stabilization to intermediates and, eventually, to the final folded structure. The process continues until complete domains form and the entire polypeptide is folded, Fig. 4-28. Notably, proteins dominated by close-range interactions between pairs of residues generally located near each other in the polypeptide sequence tend to fold faster than proteins with more complex folding patterns and with many long-range interactions between different segments. As larger proteins with multiple domains are synthesized, domains near the amino terminus, which are synthesized first, may fold before the entire polypeptide has been assembled. Thermodynamically, the folding process can be viewed as a kind of free energy funnel, Fig. 4-29. The unfolded states are characterized by a high degree of conformational entropy and relatively high free energy. As folding proceeds, the narrowing of the funnel reflects the decrease in the conformational space that must be searched as the protein approaches its native state. Small depressions along the sides of the free energy funnel represent semi-stable intermediates that can briefly slow the folding process. At the bottom of the funnel, an ensemble of folding intermediates has been reduced to a single native conformation, or one of a small set of native conformations. The funnels can have a variety of shapes, depending on the complexity of the folding pathway, the existence of semi-stable intermediates, and the potential for particular intermediates to assemble into aggregates of misfolded proteins, Fig. 4-29. Figure 4-28. A protein thermodynamic stability is not evenly distributed over the structure of a protein. The molecule has regions of relatively high stability and others of low or negligible stability. For example, a protein may have two stable domains joined by a segment that is entirely disordered. Regions of low stability may allow a protein to alter its conformation between two or more states. As we shall see in the next two chapters, variations in the stability of regions within a protein are often essential to protein function. Intrinsically disordered proteins or protein segments do not fold at all. As our understanding of protein folding and protein structure improves, increasingly sophisticated computer programs for predicting the structure of proteins from their amino acid sequence are being developed. Prediction of protein structure is a specialty field of bioinformatics, and progress in this area is monitored with a biennial test called the CASP, Critical Assessment of Structural Prediction, competition. Hundreds of research groups from around the world vie to predict the structure of an assigned protein, whose structure has been determined but not yet published. The most successful teams are invited to present their results at a CASP conference. The success of these efforts is improving rapidly, with correct predictions for smaller proteins becoming common. Figure 4. On virtually every pathway, some proteins undergo assisted folding. Not all proteins fold spontaneously as they are synthesized in the cell. Folding for many proteins requires chaperones, proteins that interact with partially folded or improperly folded polypeptides, facilitating correct folding pathways or providing microenvironments in which folding can occur. Several types of molecular chaperones are found in organisms ranging from bacteria to humans. Two major families of chaperones, both well studied, are the HSP70 family and the chaperonins. The HSP70 family of proteins generally have a molecular weight near 70,000 and are more abundant in cells stressed by elevated temperatures, hence, heat shock proteins of Mr. 70,000, or HSP70. HSP70 proteins bind to regions of unfolded polypeptides that are rich in hydrophobic residues. These chaperones thus protect both proteins subject to denaturation by heat and new peptide molecules being synthesized and not yet folded. HSP70 proteins also block the folding of certain proteins that must remain unfolded until they have been translocated across a membrane, as described in Chapter 27. Some chaperones also facilitate the quaternary assembly of oligomeric proteins. The HSP70 proteins bind to and release polypeptides in a cycle that uses energy from ATP hydrolysis and involves several other proteins, including a class called HSP40. Figure 4-30 illustrates chaperone-assisted folding as elucidated for the eukaryotic HSP70 and HSP40 chaperones. The binding of an unfolded polypeptide by an HSP70 chaperone may break up a protein aggregate or prevent the formation of a new one. When the bound polypeptide is released, it has a chance to resume folding to its native structure. If folding does not occur rapidly enough, the polypeptide may be bound again and the process repeated. Alternatively, the HSP70 bound polypeptide may be delivered to a chaperonin. Chaperonins are elaborate protein complexes required for the folding of some cellular proteins that do not fold spontaneously. In E. coli, an estimated 10% to 15% of cellular proteins require the resident chaperonin system, called GROEL/GROES, for folding under normal conditions. Up to 30% require this assistance when the cells are heat-stressed. The analogous chaperonin system in eukaryotes is called HSP60. The chaperonins first became known when they were found to be necessary for the growth of certain bacterial viruses, hence the designation GRO. These chaperone proteins are structured as a series of multi-subunit rings, forming two chambers oriented back-to-back. An unfolded protein is first bound to an exposed hydrophobic surface near the apical end of one GROEL chamber. The protein is then trapped within the chamber, when it is capped transiently by the GROES lid, Fig. 4-31. GROEL undergoes substantial conformational changes, coupled to slow ATP hydrolysis, which also regulates the binding and release of GROES. Inside the chamber, a protein has about 10 seconds to fold, the time required for the bound ATP to hydrolyze, constraining a protein within the chamber. Prevents inappropriate protein aggregation and also restricts the conformational space that a polypeptide chain can explore as it folds. The protein is released when the GROES cap dissociates, but can rebind rapidly for another round if folding has not been completed. The two chambers in a GROEL complex alternate in binding and releasing unfolded polypeptide substrates. In eukaryotes, the HSP60 system utilizes a similar process to fold proteins. However, in place of the GROES lid, protrusions from the apical domains of the subunits flex and close over the chamber. The ATP hydrolytic cycle is also slower in the HSP60 complexes, giving the constrained proteins more time to fold. Figure 4-30 chaperone 2-KHO. Finally, the folding pathways of some proteins require two enzymes that catalyze isomerization reactions. Protein disulfide isomerase, PDI, is a widely distributed enzyme that catalyzes the interchange, or shuffling, of disulfide bonds until the bonds of the native conformation are formed. Among its functions, PDI catalyzes the elimination of folding intermediates with inappropriate disulfide crosslinks. Peptide prolyl cis trans isomerase, PPI, catalyzes the interconversion of the cis and trans isomers of peptide bonds formed by pro-residues, Fig. 4-8, which can be a slow step in the folding of proteins that contain some pro-peptide bonds in the cis configuration. Defects in protein folding provide the molecular basis for a wide range of human genetic disorders. Despite the many processes that assist in protein folding, misfolding does occur. In fact, protein misfolding is a substantial problem in all cells, and a quarter or more of all polypeptides synthesized may be destroyed because they do not fold correctly. In some cases, the misfolding causes or contributes to the development of serious disease. Many conditions, including type 2 diabetes, Alzheimer's disease, Huntington disease, and Parkinson disease, are associated with a misfolding mechanism. A soluble protein that is normally secreted from the cell is secreted in a misfolded state and converted into an insoluble extracellular amyloid fiber. The diseases are collectively referred to as amyloidoses. The fibers are highly ordered and unbranched, with a diameter of 7 to 10 nanometers and a high degree of beta sheet structure. The beta segments are oriented perpendicular to the axis of the fiber. In some amyloid fibers, the overall structure includes 2 layers of beta sheet, such as that shown for amyloid beta peptide in Figure 4. 32. Many proteins can take on the amyloid fibril structure as an alternative to their normal folded conformations, and most of these proteins have a concentration of aromatic amino acid residues in a core region of beta sheet or alpha helix. The proteins are secreted in an incompletely folded conformation. The core, or some part of it, folds into a beta sheet before the rest of the protein folds correctly, and the beta sheets from 2 or more incompletely folded protein molecules associate to begin forming an amyloid fibril. The fibril grows in the extracellular space. Other parts of the protein then fold differently, remaining on the outside of the beta sheet core in the growing fibril. The effect of aromatic residues in stabilizing the structure is shown in Figure 432c. Because most of the protein molecules fold normally, the onset of symptoms in the amyloidoses is often very slow. If a person inherits a mutation such as substitution with an aromatic residue at a position that favors formation of amyloid fibrils, disease symptoms may begin at an earlier age. Figure 4. In the heptameric ring. In eukaryotes, proteins destined for secretion undergo their initial folding in the endoplasmic reticulum, see pathway in Chapter 27. When stress conditions arise, or when protein synthesis threatens to overwhelm the protein folding capacity of the unfolded proteins can accumulate. These conditions trigger the unfolded protein response, UPR. A set of transcriptional regulators that constitute the UPR bring the various systems into alignment by increasing the concentration of chaperones in the or decreasing the rate of overall protein synthesis, or both. Amyloid aggregates that form before the UPR can come into play may be removed. Some are degraded by autophagy. In this process, the aggregates are first encapsulated in a membrane, then the contents of the resulting vesicle are degraded after the vesicle docks with a cytosolic lysosome. Alternatively, misfolded proteins can be degraded by a system of proteases called the ubiquitin proteasome system described in Chapter 27. Defects in any of these systems decrease the capacity to deal with misfolded proteins and increase the propensity for development of amyloid-related diseases. The UPR is a complex response involving many protein factors and signals, and inactivation of UPR components may have positive or negative effects on the degree of protein misfolding. This system is an attractive drug target for protein misfolding, amyloid diseases. Some amyloidoses are systemic, involving many tissues. Primary systemic amyloidosis is caused by deposition of fibrils consisting of misfolded immunoglobulin light chains, see Chapter 5, or fragments of light chains derived from proteolytic degradation. The mean age of onset is about 65 years. Patients have symptoms including fatigue, hoarseness, swelling, and weight loss, and many die within the first year after diagnosis. The kidneys or heart are often the most affected organs. Some amyloidoses are associated with other types of disease. People with certain chronic infectious or inflammatory diseases such as rheumatoid arthritis, tuberculosis, cystic fibrosis, and some cancers can experience a sharp increase in secretion of an amyloid-prone polypeptide called serum amyloid A (SAA) protein. This protein, or fragments of it, deposits in the connective tissue of the spleen, kidney, and liver, and around the heart. People with this condition, known as secondary systemic amyloidosis, have a wide range of symptoms, depending on the organs initially affected. The disease is generally fatal within a few years. More than 80 amyloidoses are associated with mutations in transthyretin, a protein that binds to and transports thyroid hormones, distributing them throughout the body and brain. A variety of mutations in this protein lead to amyloid deposition concentrated around different tissues, thus producing different symptoms. Amyloidoses are also associated with inherited mutations in the protein's lysozyme, fibrinogen A-alpha chain, and apolipoproteins AI and A2. All of these proteins are described in later chapters. Figure 4 to 32 Formation of disease. Some amyloid diseases are associated with particular organs. The amyloid-prone protein is generally secreted only by the affected tissue, and its locally high concentration leads to amyloid deposition around that tissue, although some of the protein may be distributed systemically. One common site of amyloid deposition is near the pancreatic islet beta cells, responsible for insulin secretion and regulation of glucose metabolism. See Fig. 23-27. Secretion by beta cells of a small, 37 amino acid peptide called islet amyloid polypeptide, IAPP, or amylin, can lead to amyloid deposits around the islets, gradually destroying the cells. A healthy human adult has 1 to 1.5 million pancreatic beta cells. With progressive loss of these cells, glucose homeostasis is affected, and eventually, when 50% or more of the cells are lost, the condition matures into type 2, non-insulin dependent, diabetes mellitus. The amyloid deposition diseases that trigger neurodegeneration, particularly in older adults, are a special class of localized amyloidoses. Alzheimer's disease is associated with extracellular amyloid deposition by neurons, involving the amyloid beta peptide, Fig. 432b, derived from a larger transmembrane protein, amyloid beta precursor protein, found in most human tissues. When it is part of the larger protein, the peptide is composed of two alpha helical segments spanning the membrane. When the external and internal domains are cleaved off by specific proteases, the relatively unstable amyloid beta peptide leaves the membrane and loses its alpha helical structure. It can then take the form of two layers of extended parallel beta sheet, which can slowly assemble into amyloid fibrils. Fig. 432c. Deposits of these amyloid fibers seem to be the primary cause of Alzheimer's disease, but a second type of amyloid-like aggregation, involving a protein called tau, also occurs intracellularly, in neurons, in people with Alzheimer's disease. Inherited mutations in the tau protein do not result in Alzheimer's disease, but they cause a frontotemporal dementia and Parkinsonism, a condition with symptoms resembling Parkinson's disease, that can be equally devastating. Several other neurodegenerative conditions involve intracellular aggregation of misfolded proteins. In Parkinson's disease, the misfolded form of the protein alpha-synuclein aggregates into spherical filamentous masses called Lewy bodies. Huntington disease involves the protein huntingtin, which has a long polyglutamine repeat. In some individuals, the polyglutamine repeat is longer than normal, and a more subtle type of intracellular aggregation occurs. Notably, when the mutant human proteins involved in Parkinson's disease and Huntington disease are expressed in Drosophila melanogaster, the flies display neurodegeneration expressed as eye deterioration, tremors, and early death. All of these symptoms are highly suppressed if expression of the HSP70 chaperone is also increased. Box 4 to 6 Medicine Death by Misfolding. The prion diseases. A misfolded brain protein seems to be the causative agent of several rare degenerative brain diseases in mammals. Perhaps the best known of these is bovine spongiform encephalopathy, BSE, also known as mad cow disease. Related diseases include Kuru and Creutzfeldt-Jakob disease in humans, scrapie in sheep, and chronic wasting disease in deer and elk. These diseases are also referred to as spongiform encephalopathies, because the diseased brain frequently becomes riddled with holes. Fig. 1. Progressive deterioration of the brain leads to a spectrum of neurological symptoms, including weight loss, erratic behavior, problems with posture, balance, and coordination, and loss of cognitive function. The diseases are fatal. In the 1960s, investigators found that preparations of the disease-causing agents seemed to lack nucleic acids. At this time, Tikva Alper suggested that the agent was a protein. Initially, the idea seemed heretical. All disease-causing agents known up to that time, viruses, bacteria, fungi, and so on, contained nucleic acids, and their virulence was related to genetic reproduction and propagation. However, for decades of investigations, pursued most notably by Stanley Prusiner, have provided evidence that spongiform encephalopathies are different. The infectious agent has been traced to a single protein, Mr. 28,000, which Prusiner dubbed prion protein, PRP. The name was derived from Proteostasis infectious, but Prusiner thought that prion sounded better than proin. Prion protein is a normal constituent of brain tissue in all mammals. Its role is not known in detail, but it may have a molecular signaling function. Strains of mice lacking the gene for PRP, and thus the protein itself, suffer no obvious ill effects. Illness occurs only when the normal cellular PRP, or PRPC, occurs in an altered conformation called PRPSC. SC denotes scrappy. The structure of PRPC has two alpha helices. The structure of PRPSC is very different, with much of the structure converted to amyloid-like beta sheets, Fig2. The interaction of PRPSC with PRPC converts the latter to PRPSC, initiating a domino effect in which more and more of the brain protein converts to the disease-causing form. The mechanism by which the presence of PRPSC leads to spongiform encephalopathy is not understood. Figure 1 stains section of cerebral cortex from the In inherited forms of prion diseases, a mutation in the gene encoding PRP produces a change in one amino acid residue that is believed to make the conversion of PRPC to PRPSC more likely. A complete understanding of prion diseases awaits new information on how prion protein affects brain function. Structural information about PRP is beginning to provide insights into the molecular process that allows the prion proteins to interact so as to alter their conformation, Fig2. The significance of prions may extend well beyond spongiform encephalopathies. Evidence is building that prion-like proteins may be responsible for additional neurodegenerative diseases such as multiple system atrophy, MSA, a disease that resembles Parkinson's disease. Figure 2 structure of the globular domain. The misfolding PRPSC. Protein misfolding need not lead to amyloid formation to cause serious disease. For example, cystic fibrosis is caused by defects in a membrane-bound protein called cystic fibrosis transmembrane conductance regulator, CFTR, which acts as a channel for chloride ions. The most common cystic fibrosis causing mutation is the deletion of a Phe residue at position 508 in CFTR, which causes improper protein folding. Most of this protein is then degraded and its normal function is lost, see box 11-2. Many of the disease-related mutations in collagen P130 also cause defective folding. A particularly remarkable type of protein misfolding is seen in the prion diseases, box 4-6. Summary 4.4 Protein denaturation and folding. The maintenance of the steady-state collection of active cellular proteins required under a particular set of conditions called proteostasis involves an elaborate set of pathways and processes that fold, refold, and degrade polypeptide chains. The three-dimensional structure and the function of most proteins can be destroyed by denaturation, demonstrating a relationship between structure and function. Some denatured proteins can renature spontaneously to form biologically active protein, showing that tertiary structure is determined by amino acid sequence. Protein folding in cells is generally hierarchical. Initially, regions of secondary structure may form, followed by folding into motifs and domains. Large ensembles of folding intermediates are rapidly brought to a single native conformation. For many proteins, folding is facilitated by HSP70 chaperones and by chaperonins. Disulfide bond formation and the cis-trans isomerization of pro-peptide bonds are catalyzed by specific enzymes. Protein misfolding is the molecular basis of a wide range of human diseases, including the amyloidoses.